Hello and welcome back to Completely and Utterly Spore-ing, a podcast about bacterial endospore formation, where it shouldn't matter if those words don't really mean much to you. I'm still Kit, a biomedical science student and a spore enthusiast, and Callum's still here.
I'm
Hello, Callum.
you say that like I could have left from last time round. It was fun. We're ready to do some more sporing. We're ready to talk about some more spores. And yeah, I suppose this time we're actually getting into what your research is, which is very exciting.
Hopefully, we can continue to shed a bit of light on a part of microbiology that you maybe didn't know existed or particularly care existed, but we're going to talk about it anyway.
We will do. We will continue to persist to do that.
Yes. This episode will hopefully go into a lot more detail on the concept of commitment, and I guess we introduced the ideas of what those things are, and we'll go over that again. But to start to go into kind of what's going on at a genetic level, because
Everything is determined by genes at some level, pretty much.
yeah, because molecular microbiology is all about what I guess DNA as a molecule, what is that doing, and how is that controlling behavior or processes within these cells.
I mean, last episode we did talk quite a bit about the genes being activated from these different stages during the sporulation process, and so I guess we need to go into that level of detail if we're going to understand the process of creating spores.
Absolutely. So, Callum, why don't you tell me what you remember about commitment?
About commitment, well, so so there is, as far as I'm aware from last episode, for those who did tune in, so there is a bacteria state, and or not sure if state's the right word, but to complete the sporulation process pretty much. But then there is a stage throughout those 7 hours where there is essentially like a point of no return, I suppose you could describe it as, pretty much. And it's at that point that the spore is committed, to use the I guess the word that we're talking about, committed to becoming a spore, essentially. And it's obviously, we were interested in this concept of commitment because that's the you were trying to intervene in that process in some way, essentially, and seeing if it was possible for the spores to maybe turn around in essentially during this sporulation process.
Absolutely. So, commitment is actually not just for spores. It's a process, it's a term used to describe anything, any cell differentiation process. There is an idea of commitment, and there's the point of actually the cell obviously makes a decision, it's not conscious in the same way, but it makes a choice. There's a set of changes that or prerequisites that allow a process to start. But the start of the process doesn't always mean it has to finish the process, but somewhere along there, it can no longer turn around. And that's the same for things like stem cells. It's the same for any process of differentiation. And it's just, we've kind of maybe chosen spores as a model for that. Again, a lot of research in biology, we choose things that are easy to study, and then we can kind of expand out from that. Once we understand it, we can see how we can apply that to things that are maybe more dangerous to study or more complicated to study. So, for example, using bacillus rather than stem cells, they're single-celled and they are quicker to grow and they are easier to grow. It's a lot easier to work it out in a bacteria and then see what we can extend to humans or mammals, rather than starting with the mammals and going, oh, there's too much going on here. So it's kind of, model organisms are generally used, and commitment with choosing the process of sporulation as a kind of almost like a model for cell differentiation, so that we can start to understand what's going on and extend that to a load of other processes, which...
But yeah, but as you mentioned, commitment isn't something that is specific to spores, and so that means that this research is very much applicable to those, like the perhaps when studying the bacteria eventually that are more harmful and that would be useful for things like medication or treatment of diseases and with when pathogens are involved, because ultimately lots of different cells go through this, have this commitment aspect to it.
Yeah, absolutely. The term you used, you remembered, point of no return. That is the phrase that explains commitment. I would say
Thank you.
it's just everything. If Callum decides to walk up the stairs, at what point can he no longer turn around on the stairs?
No, pretty much. So it's like it is that idea of the different stages, and what point is it inevitable that it's going to reach the end stage, pretty much.
Yeah, the term decision, or the idea that they decide to sporulate, is a little bit of a sweeping statement. It's not, obviously they're not thinking, should I do this? They're feeling the environment, they are sensing things in the environment, and things are being regulated by that. So the idea that genes get turned on and off, we've talked about it before, but it sounds a bit sort of,
I think last time we used the idea of like a flow chart that you might see, say, in like algorithms in computer science. So it's not necessarily thinking, it's just that flow chart diagram of, right, are we experiencing this in the environment? Yes or no? If we are experiencing this, like maybe, as we mentioned, the lack of like food or nutrients for the bacteria, then as part of that flow chart, it enters into this sporulation state. And so it's not really decision making, it's more just following that flow chart. And then it's just saying, at what point in this flow chart is it always going to end up in this end state of a spore?
Yeah, and the way we talk about it, you learn in biology pretty early on that everything is determined by a signaling pathway of some sort, and it's just sort of, it detects this, it'll make that, and then that'll make that. And the idea that it has to complete sporulation, the concept there is less that it will absolutely definitely finish, it's more that it can't do anything else. So if it had, for example, if it had a problem with step 7, it would die rather than go back to a normal cell.
It's more, commitment is, it's more about the fact that it can't reverse what it's just done.
Yes, yeah. So it's obligated to complete the process. And if it can't complete the process, it has no other options. Rather than earlier on, if you thought, you know what, I might go up the stairs, and then you saw the stairs and you thought, no, the stairs are on fire, I'm not going to walk up that. There's a choice there, but once you're on the stairs and you go, ow, actually I'm burning my feet, you're like, oh, I'm halfway up the stairs, I'm going to have to walk the same amount on the fire either way. You're not necessarily going to turn around.
It's a state where you can't go back to the original state you were just in, and that's exactly the case for these spores.
So I talked about the idea of signaling pathways and the idea of turning genes on and off, and I talked about earlier, I said about, in last episode I talked about the extension leads and the idea that we turn off, to mutate something, we turn off one thing that is responsible for like 200 things. We're going to get a little bit more science in here. I do apologize.
Oh, I'm ready. I'm ready. This is gonna. We might have a few questions coming after this, but we'll give it a go.
We're starting the concept. This is again not unique to bacteria and not unique to sporulation, not unique to anything like that. It is how genetics works in general. It's slightly simpler in bacteria than it is in multi-celled organisms because of the fact that there's slightly less interacting factors. The genetics and the organization systems are slightly simpler in bacteria, so it's easier. But you have something called a regulon, and this is a group of genes that is controlled by another gene. So if a gene is turned on, it makes a protein, or it makes another molecule off its instructions. It's basically revealing instructions. It's not the gene itself isn't a protein. It is the instructions to make something. So when you have a regulon is turned on, it's because a gene has made a protein that can then activate the site for all of the other genes. So for example, it's called a regulon, and then it's called an operon. So you have a a small molecule that can then bind to the start of a gene so that the thing that reads it knows where to look. It's almost like a bookmark.
Okay.
Or like someone holding a page with their hand when a book is shut, and it's like telling you where to open to so that you can start reading. Because otherwise it doesn't know where to bind. So you have like reading, almost like a reading protein that starts to make the proteins, and it can only attach where it's allowed to, I guess. So when genes are turned off, something is blocking the binding site for this reading complex. And so it can't start making something. But when you have a gene that is responsible for turning on an entire regulon, so for an entire group of genes, they're called sigma factors, and it's a small molecule, like a protein, that can allow binding to start reading of however many genes it is responsible for. So we have a cascade of different sigma factors in the sporulation process, and in all metabolic processes, really, differentiation processes, where, for example, every time you're turning on 100 genes, you're turning on a gene that can then turn on another 100, maybe.
Yeah, so
you've got these.
In your analogy, the regulons are the group of genes that are, I guess you could say, turned on, to use I guess just non-specialist terminology.
Yeah.
Regulons are the group of genes that are turned on by the activation of other genes, and it's these sigma factors which basically, in a way, allow that to happen, essentially.
Yeah. So when I said about step 2 and step 3, what we would actually be talking about there is when you have your big cell and your small cell, the first sigma factor, sigma F, is turned on in the small cell, and then a series of changes happens, and then in the big cell, sigma E turns on, and then sigma G turns on in the small cell, and then sigma K turns on in the big cell again. So you've got this kind of feedback from one to the other repeatedly to get this series of steps. But we know that something turned on by sigma F, but before sigma E, is the point of no return, is the point where it can't turn around anymore. If we delete some of the genes under sigma F, but before sigma E, it won't be able to it will be able to turn around. The mutants that we worked with was quite standard. If you're trying to study commitment, deleting sigma E traps the cell because it can't go past the septum stage. So it has the line forms, and because it doesn't have sigma E, which it needs for engulfment, it just stays there and dies, or stays there and starts to uncommit. So you're creating an environment by deleting sigma E, you're creating an environment where it has to try and reverse, or it will die. So it's sort of like a forced.
It's like when you're saying you're backing it into a corner, you're giving it no other decision other than you have to turn back, or otherwise you will die. And that's what you're doing by deleting this sigma E factor. Oh, that's really interesting. Okay, yeah.
And you can see just how important in sigma E mutants, you can see just how important that next step is to it, because these mutants, normally when a cell starts to form a spore, the first few steps, it forms a line, and then it forms like there's the little cell and the big cell. In a sigma E mutant, the first septum, it forms a line, and that fails, it can't engulf, so it tries again on the other end.
Oh, what? So do you mean the small cell tries to engulf the mother cell? Is that what you mean?
No, 2 small cells form.
Oh, I see.
So on each end, you've got like a.
Oh, right. Yeah.
It forms, so when normal sporulating cells will have that there's one small cell, one big cell. When we have none of the ones that I looked at under a microscope looked like this. All of mine had like a rectangle and then 2 semicircles on each end, because of the fact that it tries to form one spore, that fails, it tries again because it went right. No, that went wrong. We need to be able to do this. Let's try again. And then you end up with an interesting scenario because they replicate the DNA. So there's 2 sets of DNA and 3 compartments
Oh, okay.
So
So there hasn't. So there's not. Yeah, because I remember you said this is. By the way, this is so interesting because I've seen the photos that you showed me when you were doing this of under the microscope, and now I'm actually understanding it. Yeah, because last time we were talking about how was it was it 100 out of the two sets of DNA, which I suppose you could say makes up like 200% of DNA because it's obviously two copies. It
yes,
was like
yeah,
it was about 170% and a 30% split. But
yes,
now for these spores that have the sigma E factor deleted, that instead of being shared between the mother cell and the baby cell, I can't remember, it's the smaller cell. It's not a baby cell. It's now
no,
split between a mother cell and two smaller cells.
yes. So it's
Ah,
still,
okay.
because of the fact that sigma E is deleted, but everything else is before that is functional. It still has the ability to move the DNA into the small cell. But what happens is you get the kind of 30% in the small cell on one side and 30% in the small cell on the other side, and then they successfully transport. You've then got two small cells with a set of DNA in it, and the middle section, the mother cell, is empty. It's got no DNA in it, and it just dies.
Wait, but okay, so wait, but what happened? So you mentioned 30% for either cell. What happened to the other 140%?
That is to start off with in the mother cell, and then
Yeah.
because of the systems, because there's something called there's a gene called SPO2E, and it just means sporulation step 2E, and it means that
Okay.
it it's a translocation system. It makes a set of like a almost like a special tube or like movement device that puts the DNA that originally is trapped in between the two cells, pushes it all into the small cell. But
Right.
because of the
Okay.
fact that it's doing it twice, because of the fact that it's trying twice, it tries with both sets of DNA, and then you leave the middle, the mother cell, without any DNA. So obviously it cannot finish, it can't do any of the next steps because the next step in engulfment involves sigma E in the mother cell, which no longer has any DNA.
Yeah. Okay.
So it's just an empty box, to be
And
honest.
those and those and the two sets of DNA being transferred to the two smaller cells, is that split evenly? So do they each now have like a complete set of DNA?
Yes, they do. And these are
Okay.
the places. So what we're trying to do is in this sigma E mutant, you've formed a cell that has started to sporulate. It's failed. It's tried again, and it's got now. You get to a point. It's if it can't reverse, then its end point is two small cells with DNA in them and an empty cell, and all three will die because they can't do anything else. But if they can reverse the small cells, you've got a cell on each end that is small but it's complete in its DNA. It's got all of the information it needs to start surviving and replicating like a normal cell. It's just not turned on, or it's not
Right.
allowed
Okay.
to.
So this is because this would have been my next question: is if the two sets of smaller cells that survive have complete sets of DNA, how come they might also die? But you're saying this might be because of the fact that they've got certain genes turned off that they need to essentially survive and reproduce like a normal cell.
Yes, we're going
Okay.
back to sigma factors. So
Okay.
there
Okay.
is not to return to the sigma factors too quickly, but
Yeah.
um,
Yeah.
so in the sigma F is the first sigma factor for the four spores sporulation process, and it turns on the things that are involved in commitment that we know are involved in commitment. Because if you delete sigma F, the small cells can just turn around, no problem, and start
Yeah.
replicating.
Yeah.
But
Because
if
we
you have
said it's in
sigma F
between.
on, they can't.
Yeah, but we said it's in between F and E, wasn't it? That this process
Yes,
of commitment happens. Yeah.
yes. So the difference between these two mutants is if sigma F is deleted, you have the DNA goes into the small cells, and then they start growing out like normal cells. But if you have
Oh,
sigma E
okay.
deleted, you have that it goes into the small cells and then the whole thing dies. Because in
Oh,
sigma F,
right.
it can turn around. In sigma E, it gets stuck. So that's the difference. The reason why sigma, again, we're now going back into the kind of theory rather than fact, because this is an active area of research. But sigma factors, there are a number of sigma factors in a cascade involved in sporulation, but in normal vegetative growth is what we call it, the kind of healthy cells, there is a sigma factor called sigma A, and this is referred to as the housekeeping regulon because it turns on all of the genes you need for normal metabolism and healthy activity.
Okay.
Because of the fact that sigma F is on in the spore, sigma A shouldn't be. They are responsible for two different metabolisms, and it's not really the most sensible to have two metabolisms on at once because one is for surviving and one is for drying yourself out and starting to just stop doing everything. So the idea of stopping metabolism and maintaining metabolism, it's a bit unlikely that these could happen at the same time. But you kind of need them. One of the things that we were trying to do in my research was have them on at the same time and see what happened, and it created some quite strange mutants.
On a completely separate, like, just to create an analogy, I remember like hearing something. This is gonna like seem like it's just completely unrelated, but in psychology, you're told that like one way to I guess help with a phobia is to actively try and relax, and that's because they're completely two opposite things. And if one is happening, then the other can't happen. And it's a little bit like, I think from what I'm hearing, these two sigma factors, because as you mentioned, they've got two different, not different, but just in a way opposite types of metabolisms. Because as you mentioned, one is for healthy growth and one is for obviously surviving, which is what happens when the sporulation process happens. Because they're doing completely opposite things, it seems in theory that they can't do anything. Just out of interest, a quick question. Is this, you mentioned that when you delete the sigma F factor, the cell is able to reverse, and then but then if you delete the sigma E factor, then it's the two smaller cells die. Is that how researchers found out what sigma factor or what stage in this sporulation process commitment happens?
Yes, exactly.
Okay,
it's the fact that it gets stuck. We know that step 2, if you get rid of the step 2 part, it can turn around fine. And if you get rid of the step 3 part, it dies. So it must be somewhere in between step 2 and step 3 that it gets stuck. Because the thing is, turning on sigma F turns on 100 things. It's not that the whole 100 things are what stops it. It's some of the 100 things. Because we know there's so that's why we know it's not sigma F specifically. It's something under sigma F. So it's like if I turn off an extension lead, my lava lamp turns off. It's not, if I unplug the lava lamp, the same effect happens. So we're trying to get more specific with it so that we know exactly what it is, rather than just, like if I said, I want to turn my oven off, I'll just cause a power cut. It's a little bit...
You've got multiple different options that you can do along that process which have the same outcome. So it's just you're essentially narrowing. So there's only 100 genes to choose from after that.
Only issue there is it's not that one of those 100 genes, it's that an unknown combination of those 100 genes, and we don't know how many genes and how they interact with each other. So it might be that it's 5 genes together and some of them don't have an effect until they're put together. So this is why it can be difficult. It's hard to reverse. It's hard to reverse engineer. One of the things the PhD student that I was supervised by had been doing some trial and error where they'd picked specific genes and deleted them, and then measured how good at outgrowing things were, and then identified that there were a few different things that caused a little change, and then if you combine them together, there was what she considered the most uncommitted mutant, which was a combination of 3 genes, and it led to like an amount of outgrowth, but not the full, not the when you delete sigma F, you have full outgrowth. You have that the little cell gets longer and forms lines down the middle to divide, and it's replicating. It's completely happy. It's able to do things. You have, when there's limited outgrowth, you might have that some of the cells grow a bit longer, and some of them can start to divide, but it's a much lower proportion. And that's why it's considered partial uncommitment, because you know that it's nowhere near the level you see when you delete sigma F.
Yeah, because you've got I guess the process in all these different stages, you have sigma F and then it's sigma F that turns on all of these different genes. So if you delete sigma F, what you're essentially doing is you're not deleting the genes under sigma F, but you're stopping them from activating. So that means that all of the genes that are required for this commitment process, all of them are turned off. And so that means it's absolutely fine to reverse the process. But when you keep sigma F on, but then you delete certain genes, it might be that some genes that you're deleting were involved in the commitment process, but then you might be leaving others that are, so it can still be partially committed, essentially, is what you're saying.
Yeah. So the way that we measure, I said before about the fact that there was a way to quantify outgrowth and a way to quantify commitment as a concept.
Yeah.
So I said that there were three things that you could measure. So the questions are, is it bigger? Is it able to replicate its DNA? And does it have sigma A activity?
Right,
and now that sounds, when I say does it have sigma A activity, you're probably thinking, but it's got sigma F. It can't have sigma A activity. That we just explained why that didn't make sense logically. But if you've deleted the things under sigma F that oppose sigma A, you can have sigma A and sigma F on, hypothetically, at the same time. But this is all, again, this is an active area of research where we're trying to do things and we don't fully, that none of this is confirmed or positively known. I'm also not allowed to tell you the name of the genes because they're not published yet.
Right. Oh, interesting. Okay, we've got some top secret information going on here.
Yes.
On completely that's Spore-ing. No, but it makes sense though, because you're not talking about genes that are fully committed to this Spore-ing process. You're talking about those that are partially committed. And so if some of these, so just some of these genes that are might be related to this commitment process, because I suppose if you're talking about this commitment process, parts of that would involve suppressing or stopping sigma A from occurring, to use non-specialist terminology. But because if you're turning into a spore, you don't want sigma A to be happening because there's not the nutrients and the conditions available to complete normal metabolism. So that will be, part of these genes under sigma F is suppressing sigma A. And so if you don't have them, if you delete those genes, then you can have sigma A occurring, even though it technically opposes sigma F function.
Yes, because again, this is the idea that deleting sigma F is like a hard reset. It's not actually solving the problem. It's just solving the thing before the problem so that it doesn't happen.
Yeah.
So it's the equivalent of an IT person telling you to turn it off and on again. It's we're doing something, we're going a few steps back because it's much easier than finding out the actual things. But now we understand the process more. The next logical step is to find out those actual things, because we need to know, rather than just knowing, oh, well, if we reset it, it'll just go back to normal. Knowing what the actual problem was is a lot more useful for our understanding of cell differentiation as a concept and our understanding of commitment as a concept, not just for bacteria, but to hopefully translate to things like stem cells and cancer in mammals, and potentially also then for understanding these pathogens that are undergoing sporulation in order to evade our treatment of them, because that's quite inconvenient for people trying not to be ill.
At the end of the day, you just understanding that sigma F, if you turn that off, then they can reverse. That's not the ultimate end goal if you're wanting to, because you need to, I think, as you mentioned, I think what you're saying is that you need to understand it at a deeper level if you want to apply it to other cells, because you need to understand the exact process. And so that's why we're not just content with understanding, right, okay, sigma F, if we delete that, then the process can reverse. We want to know the exact genes that are of these 100 genes under sigma F that are involved in this commitment process.
Yes, and the reason why that is because if we understand what the genes are, for example, if there are 5 genes that in combination completely uncommit a cell when deleted, we can find out what those genes are doing and then understand why they uncommit. a cell if deleted. So for example, like if one of the genes is to do with the membrane and one of the genes is to do with movement, or one of the genes is to a protein that protects the DNA or something like that, you know, okay, this is committing it. You can start to think not only these are the genes that commit a cell to sporulation, rather these are the genes that commit a cell to sporulation because they make these choices, or because they put them, they start off this pathway. And that is what we actually want to know, rather than just, oh, we delete those and it goes backwards. We've solved it. It's more, we delete those and it goes backwards. Why?
Yeah. Because I think last time you mentioned like the reason that spores are, if like say for example like some pathogens undergo sporulation, it's very difficult to fight off spores because they're so resistant to everything. They've got this hardened cell wall essentially. And so if we could, for example, find what particular gene in that like under sigma F that is responsible, or what particular set of genes is responsible for this hardened cell wall, then again, theoretically, am I right in saying that you could delete those for some certain pathogens, and that means that it would be easier to fight off the pathogens inside someone?
That is a little bit the problem. The formation of the wall and things like that are much later in the process. They happen after engulfment. The mother cell helps produce those way later. And also, you wouldn't. The issue with the idea of deleting it in a pathogen, you can only delete it in the pathogen samples that you have. You can't just delete it in all of them.
That's true. Yeah,
is the issue. So instead of trying to delete it, kind of like to like round up all of the pathogens and like cut their noses off almost. You can't. It's slightly unrealistic, but potentially understanding that commitment is, or understanding that the thing that starts off this process is a certain factor, creating a molecule or a chemical that interferes with that step, we could prevent successful spores from forming in the first place.
Okay.
So, for example, if you knew you had, again, this is sort of a very idyllic view, the idea that once we understand spores fully, we could create medication that forces them to germinate and prevents them from forming spores. So if someone had spores inside them, you could force them to start growing again so that they could be treated. And if someone had someone had a pathogen, you could prevent them from forming spores by giving them a medication that maybe prevented that process from starting.
Yeah.
Again, this is hypothetical. This is fully just, in a perfect world would be able to do this. But the more we understand about these processes, the more we understand if something like that is actually possible.
So, you need to find out if it's possible, and if so, it's decades of research away potentially.
Yes. Yeah. No, we're still on what does this gene do? If I turn it off, does it still live?
Yes, it's still an important step to do, and it's still it's still fascinating in its own right.
But it feels more like obviously it has applications, but at the stage that we are, it feels more like interest than application. Like you said about, it's the finding out stage, not the applying stage. We understand some things, but we're not necessarily, there are some things that we still just don't understand at all. Even like we have like theory. Well, even like how in physics you have theories and then they get disproved.
Yeah.
We're still looking at there are things that we're still trying to work out that are, I guess, further back than a lot of physics things, which are a lot more into the application stage. You see, from what I hear from the people that I talk to that do physics, you're learning to apply things whilst we're still trying to work out what some things do in a lot of microbiology.
I think there's always different areas of it. So like, for example, in physics, there'll be some areas where you have a very good understanding and you're into applications, but then you can always, I think, go further in your understanding. And I'm sure that's the case for many areas of biology where you can maybe start to apply what you've learned and then also try and go a bit further with it. But I think definitely I was actually thinking of very like I won't go into specific examples from physics because the podcast is unfortunately strictly biology, but I can think of many different examples in physics where just because something, when you're doing some research, you have no, when you're doing the research and you're looking at the research beforehand, because you don't know what the results are going to be, there's no way of telling you at that stage what the applications are going to be. So that's why you almost have to do research for research's sake, or just ask questions and try and find out the answer, because you can never know how broad the applications are going to be for whatever you discover.
This is something that really excites me about academic research, or like university-based research rather than pharmaceutical-based research.
Yeah.
Because it's the idea that you're finding out to understand rather than to, there's not the end goal. Like the brilliant thing about that you did the same, well, you didn't do the same project, but you did also do an undergraduate research project over the summer. You go into it without, you have a kind of idyllic endpoint. You have a research question, but you're not necessarily expected to answer it, and you're not expected to keep it the same the entire time if things are allowed to change. The idea of doing academic research, and you have maybe an objective, but the objective is to understand, not to find the specific thing that will fix this problem. It's just, we need to know more about this thing. Can you go and have a look, please? Rather than, we need the solution to this problem.
Yeah, and that's something I found out actually about while doing this project, and actually I think is what a lot of people don't know is that's actually what happens most of the time with PhD research, for example, or like completing your thesis, or even further on than that, with just if you're a professor doing research. It's not, when you start out to research something, you're really not looking at a specific question. You're just looking to understand something. And then once you finish the research, you think, right, what's the best story I can tell with what I found out, and that's then what you write the paper on. Because often when you read papers, it's like, often when you read research papers, it's like, oh, we started out with this specific goal, and we therefore found this, and this is our results. But that's just the way of wording it in hindsight. That's never how you start out. And that's why often the research question and the research objective can change very, very often, like more often than not for PhD research and other research, because you don't know what you're going to find, and you don't know what's going to be the most interesting from what you're understanding. Anyway, we've gone majorly off topic, but
to bring back the topic, I guess, a little bit. The title of my research project was Investigating Genes Responsible for Commitment to Sporulation in a Model Organism. It's quite a broad one. And I think that was brilliant to have something quite so broad of just the word we're just investigating. We're looking at commitment, and we're seeing what we can find out in 6 weeks from where the lab that I was working with and the PhD student that I was supervised by was currently in their progress. What can I do in 6 weeks to try and understand whatever we can a little bit more? And I think that was a brilliant experience, being thrown in. You're thrown in a little bit of the deep end because someone's going, all right, at the end of this week I found out this thing that's slightly interesting, maybe we should start looking at that. And it's the idea that it feels so like you're exploring. It's an adventure rather than, it's about the journey, not the destination, which is I hate the phrase, but it's true. It's true.
Yeah. It's a cliche phrase, but unfortunately it is actually quite applicable in this sense.
Yeah.
But no, it's definitely a good research question. I think for a topic such as URSS, it's you need it to be broad, because it's just it is just exploration at that point. And so, but yeah, certainly. I mean, I know for certain that you had a good time with it, because every time, well, you would often be very excited when I would come home to talk to you about it. So now that we've gone into proper detail with it, and we've gotten to the end of gotten to the end of 2 episodes, and we now understand the research title, and so now we can go into Steve. stick with us.
Steve has written again, what's the point of this? We'll get there. You'll find out soon, in due course.
To be fair, I think we've already gone to some quite good reasons as to why it's this is very applicable to lots of different aspects of biology and why it is important to understand. But
yeah,
Steve, if you need more of a reason, we're getting there.
Yeah.
So yeah, just stick with us.
Yeah, we'll see you, Steve. We'll see you next time, where we talk about how we're talking about all these concepts of, we can test how uncommitted this is. We can mutate this. How am I doing that in a lab? You'll have to find out, Steve.
See you next time.
See you next time.
Goodbye.